Hello Year 12. So today we're going to start looking at the nervous system. So we're going to be looking at the different types of neurons and their structure. We're going to be comparing the different types. We're going to be looking at something called myelination and what that is and why it's important. And then we're going to look at the sort of outline of the layout of the nervous system and look at the sympathetic and parasympathetic nervous system. So there's quite a bit of content in this lesson. Some of this is split between different areas of your textbook, and you'll come back to it again when you do the brain in terms of the layout of the nervous system, but we're going to come into it today as well. So just to give you a little bit of evolutionary history of the nervous system. So nervous systems allow coordination. They tend to be faster than hormonal systems, but they've evolved probably after hormonal communication or chemical signalling. And in fact, nervous system is just a modification of chemical signalling. It's still chemical signalling between cells at the end of the day. So if we think about organisms, very simple organisms, things like hydra and jellyfish and starfish, they have what's called a radial symmetry, so you can sort of slice them up, or sometimes have no symmetry at all. And these organisms can have very, very, very simple nervous coordination, which is called a neural net, but they don't have a brain. They don't have a central coordinator, or they do have regions where clumps of neurons may come together and form synapses, which you might remember from GCSE, but they don't have a brain. Now, where you start to get the formation of a head, and you start to get what's called bilateral symmetry, which is left-right symmetry, and cephalization, which is the formation of a head, then you start to get brain-like structures developing. Okay, so brain-like structures at the anterior end, so the head end of the organism, nearest to where the organism is going to experience the environment first, tends to be where the brain is. So they have a lot of sensory receptors at the head end. Okay, here's some sensory receptors. Okay, being pointed out to you. All right, on these amazing specimens of humanity, and obviously the head end is generally where you experience the environment first, so it's sensible to have your receptors at that end and close to the coordinator close by. Okay, so we're going to look at neuron structure first, and you may well recognise some of these pictures from GCSE. So we've got three types of neurons we need to be aware of. We've got motor neurons, we've got interneurons, something called relay neurons, generally what they're generally called, and sensory neurons. All right, I just want to have a look at these and go, right, well, what's, especially with the motor neuron and sensory neuron, what do you notice is similar, what do you notice is different about them? Okay, just give yourself a minute, just have a good look at these pictures. Okay, so hopefully you spotted some things like you've got this structure called the axon on both of them. Okay, you have got this green stuff around the outside here called the myelin sheath. We'll talk about that in a minute. You've got a cell body on both of them. You've got branching ends at each end of the neuron, which are called dendrites. The sensory neuron has this extra thing called a dendron. You'll notice the cell body, where most of the cytoplasm and the nucleus is slightly different positions. The axon itself and the axon or dendron are actually just projections of the cell membrane with a bit of cytoplasm running through. So this is all part of the cell. It's not a solid thing, it's a hollow tube, if you like, full of cytoplasm, and it branches at ends. So you've got lots and lots of branches, okay, and at either end. So that means it can make multiple connections at either end. Okay, so thinking about motor neurons, so motor neurons' function is to carry nerve impulses, which we're going to talk about as action potentials, because that's what they are, and they're propagated or travel along from this end, from where the cell body is, down the axon. So axons always go away from the cell body, axons away from the cell body, and they're carrying the action potential down the axon to this end, and this is where you'd have effectors. This is where muscles and glands, so different types of muscle, like skeletal muscle, which is voluntary, smooth muscle, like you have around your blood vessels and your digestive system, cardiac muscle, and also all your glands that do secretions, okay, that are attached to the nervous system. All right, so this part of the neuron will be in the central nervous system. Okay, so these will be connecting with these relay neurons here. All right, so these dendrites will be having synapses with relay neurons. Sensory neurons. Now this diagram's slightly not to scale, and actually none of this is to scale. So the sensory neurons will be transmitting action potentials from this end, and here we have receptors. So you have things like pressure receptors, like the posterior corpuscle, or you might have sensory receptors in your eye, like your rods and cones. We'll be making synapses here and making connections, and action potentials will be transmitting down along the dendron, okay, past the cell body, and then along the axon again, running away from the cell body, and this will be going into the central nervous system. Now the key thing here, this looks sort of equal either side, and actually the dendron is really long, and this is, if you think about it coming from your fingertips, a dendron might run all the way from your fingertips to your spinal cord, and just before the spinal cord there will be the cell body in a structure called a ganglion, which is just outside the central nervous system, and then the axon is actually quite short and makes synapses with relay neurons in the central nervous system. Whereas a motor neuron, the cell body is actually inside the central nervous system. So for example, it might be in the spinal cord, and then the axon comes out from the central nervous system, and so the axon is very long. So the axon here might be pretty much as long as a dendron, for example. You think they can be meters long. Think about coming from your leg going towards your spinal cord, for example, or coming from your spinal cord going towards down your leg towards your feet, whatever it is, whatever muscle it's going to attach to. Okay, these motor neurons can be very long, and so can these dendrons. So these axons here, not drawn to scale at all. In fact, also this myelin sheath, these green structures here, this is one cell, this green structure wrapped around the axon, and they're only about maybe about a millimetre. They're quite big cells, but they're about a millimetre long, so there'll be thousands of these along one neuron, or at least hundreds of them. Okay, and that dotting there showing you that it's oversimplified, but this is again wrong, because this bit should be dotted, because the dendron is a long part of the sensory neuron, not the axon. Okay, notice that there is no dendron on a motor neuron, and the dendrites actually arise from the cell body, so they sort of come out from the cell body, whereas they don't come from the cell body on a sensory neuron. They come from the ends of the dendron or the axon. Relay neurons, you'll notice relay neurons or interneurons don't have a myelin sheath around them. They're found inside the central nervous system in a region called the grey matter, so in your brain and in your spinal cord. There is myelin, and we'll talk about this in a moment, but it's nowhere near as complexly around the neurons as these are. Okay, so use your textbook, you've got a worksheet, fill in the worksheet as much as you can with information on the different types of neuron, what they do, where they transmit nerve impulses from and to, etc. Give yourself a minute on that. Oh, more than a minute, maybe 10 minutes. Okay. Now, what I like you to do is just a little task, see what you can remember now. So have a read of your sheet, and then write down at least 3 comparison points to compare the structure of sensory and motor neurons. Give yourself maximum 5 minutes. Okay, so hopefully you got the idea that motor neurons have the cell body at the end of the neuron, whereas it's at the side in the sensory neuron. They both have axons, but the motor neurons have the long axons, sensory neurons have short ones. Only sensory neurons have dendrons, or you could have written motor neurons don't have dendrons. Axon arises from the cell body on the motor neurons, but it doesn't on the sensory neuron. It's sort of the dendron and axon basically merge into each other. The motor neuron in the cell body is inside the central nervous system in the central neuron, but it's in these structures called ganglia outside the central nervous system. Both have dendrites, but the dendrites come from the cell, arise from the cell body on the motor neuron, but you don't have any on the sensory neuron, because on the cell body of the sensory neuron. Now, if you wrote this one, sensory neurons carry action potentials from receptors to the CNS, whereas motor neurons carry action potentials from the CNS to the effectors, then that's wrong, okay, because it's not a structural comparison, that's functional. Okay, maybe take a couple of minutes just to jot down anything you missed there or improve your notes on that. So let's have a look at the myelin sheath. So the myelin sheath is it develops actually as you grow, and humans when they're born, their myelin sheath is not, as far as I'm aware, is not fully formed. One of the reasons why you can't walk fully when you're born. Don't quote me on that. But the key thing is what it is. These are specialised cells that grow around the axon, and you can see here they develop and they wrap around the axon, a bit like sort of kitchen paper around the central cardboard bit of a kitchen roll, except this is a cell with layer of cell membrane, and inside that is a layer of fatty material which is called myelin. So these cells are called Schwann cells, and each cell is, well, depending on where you look, 1 to 3 micrometers long, and then, or some places say they're smaller than that, but then in between there are little gaps which are only about 1 micrometer wide, called nodes of Ranvier. And the little gaps are where this cell membrane is exposed, and all the rest of it is insulated. Myelin is electrically insulated, so wherever you get the myelin sheath, it means you actually can't have any electrical change, and that affects how nerve impulses travel along myelinated neurons. And you only get these action potential things, these changes in charge, which we'll look at later on, at the nodes of Ranvier. And that means when you look at the transmission of the action potential along a myelinated neuron, it looks like the nerve impulse or the action potential is jumping between the nodes of Ranvier, and that is called saltatory conduction, which comes, derivation of it means to jump. Saltatory is to jump, apparently. So all your motor neurons attaching to your muscles and most of your sensory neurons are myelinated, because they're transmitting nerve impulses quickly either from your receptors to the central nervous system or from your central nervous system to your skeletal muscle mainly, directly to them, and it needs to be rapid. Whereas in other parts of your nervous system you have neurons that are not as heavily myelinated. In fact, they have less, the Schwann cells that aren't growing around them as much, or they're not myelinated at all. So actually within your body you have really heavily myelinated neurons, you have lightly myelinated neurons, and then you have some that are what are called non-myelinated neurons. Now that's a bit of a misnomer in that there is myelin there are cells, but they sort of grow around the axons in a diffuse way, they're not wrapped around it, so we can talk about them as unmyelinated. And in your automatic, or what we call the autonomic nervous system, you have some unmyelinated neurons, and actually if you look in your textbook in section 13.6 you will see that, and actually we'll look at that later on. So what's the impact of myelination? Well, let's have a look here at this table. Okay, it says compare the effect of myelination and diameter on the speed of nervous conduction. So when you have a look at this, just have 2 minutes looking at it, make some comparisons. Okay, so hopefully you can look here, when you're making comparisons you have to compare sort of like with like if you're going to compare the diameter. So if we look at here, we've got crab and squid and cat and frog. So crab and squid have got these non-myelinated neurons, and they don't have any myelinated neurons. Then we've got looking at some myelinated neurons in a mammal and an amphibian. Okay, and you can see, so let's look at diameter, the effective diameter. So here we've got narrower and wider diameter axons, And you can see here, the wider the axon, the faster the speed of conduction. Now you might expect that, because if it's narrow, think about a wire. Narrow wires have greater resistance, thicker wires have lower resistance, so it's similar here. It's not the same in terms of how the conduction is occurring, but generally there's less resistance to the action potential the wider it is. And actually in the myelinated ones you can see that too. The wider axon has a faster speed of conduction here. Remember, you cannot compare, say for example, the frog and the crab for diameter, because you've got two different things here. Now the key thing to notice though is, what do we notice about the myelinated ones is myelinated neurons, the speed of conduction is much faster. That saltatory conduction, that jumping conduction, is much faster. So it allows more information to be sent more quickly. It can allow quicker responses. So where you need fast responses to stimuli, you tend to have myelinated neurons. Where you can have slower, more long-term responses, you can have non-myelinated neurons within the human nervous system. Okay, and actually you can see the significance here. This, let's sort of take sort of similar ones. This one's 20 micrometers in diameter, this one's 30, and you can see a significant increase, more than 20 times faster, just for even though with myelination, even though the diameter is actually narrower. And the whole point is you can therefore have more. You can also have more axons in a smaller space and have them work fast as well. So it's sort of also good for sort of density, if you like, being able to pack them in. Okay, so what I want you to do now, I'm going to put a load of questions up on the next slide. I want you to make sure that you can answer all of these. You can either add it to your diagrams of the nervous system layout, which are on the other worksheet that I've sent you, or make your own bullet points, however you want to do it. Give yourself sort of 15 minutes on this, and then come back and I'll pick up on some things. Okay, so hopefully you've got some sort of, you looked at this diagram. So let's think about these different things. So you've got the central nervous system and the peripheral nervous system. So central nervous system, this is your main coordinator. This is where your brain and spinal cord is, okay? And this is the bit that's taking nerve impulses, action potentials from the sensory neurons and coordinating the response, and then sending action potentials down the motor neurons to the effectors. So basically it's the link between the afferent system and the efferent system. So the peripheral nervous system is all your neurons and receptors, and effectively the effectors that link into the central nervous system, outside the central nervous system, so outside the brain, outside the spinal cord. Okay, so afferent, always remember afferent is like arriving. A for arrive, A for afferent. So this is your sensory neurons will be coming in and going into the central nervous system. The efferent like exit, okay, so egress, escape, coming away from the central nervous system. So efferent is your motor system. This can be split up into what's called your somatic system. So somatic is the bit that you have conscious control over. Okay, so you voluntarily decide to do something. So you move a muscle, raise your arm, wave at someone, okay, whatever it might be, run away. That is under your conscious control, and that will be sending nerve impulses down the motor neurons mainly to your skeletal muscles. Okay, so this is about sort of conscious movement. But you've also got the autonomic nervous system, which is the sort of involuntary system. So it's working under subconscious control. You don't have to think about it. So this is the bit that controls heart rate and breathing and digestion and everything that's going on inside your body that you don't want to have to think about to make it work. And this is all part of the motor system. So always keep that in mind. This is all part of the motor, sending things to effectors. But the effectors here tend to be a bit different. Things like your smooth muscle in your blood vessels, in your digestive system, cardiac muscle, so the heart, and also glands. Okay, and this is subdivided up into the sympathetic and the parasympathetic nervous system. All right. Now, just because this is autonomic doesn't mean you can't consciously control it. You can control your breathing. You can, to some people, can affect their heart rate. But as soon as you stop thinking about it, the automatic system will kick back in. So the central nervous system will be controlling the autonomic nervous system. Remember, they're all linked. Afferent going into central nervous system, efferent coming out of central nervous system. Okay. Remember, you've got about 86 billion neurons in your central nervous system. Most of these, remember, are your relay neurons, mostly unmyelinated. Okay, and they sort of make up your grey matter, your little grey cells, if you've ever watched any Hercule Poirot. Okay, let's have a quick look at the sympathetic and parasympathetic nervous system and their layout. So here we've got parasympathetic and sympathetic. We've got similar organs lined up here. Now the first thing you'll notice is this diagrammatic representation is also showing you the sort of layout of it, in that the neurons, the motor neurons coming out from the central nervous system in the parasympathetic system have really long axons, and then there'll be a short bundle of connections here called ganglia with short neurons running to the effectors. You've got this huge nerve called the vagus. Vagus comes from the word like vagrant, which means wanderer. It's this big nerve with lots and lots and lots of motor neurons in it that travel to lots of different organs. And then there'll be a structure called a ganglion, which is again this bundle of really sort of cell bodies, where very short motor neurons then come from there to the directly to the organs. There's always two neurons. So in the somatic nervous system, there's only one neuron going from the central nervous system to the muscle, whereas here there will always be two. There'll be the neurons coming out, and then the short ganglion, and then a short post-ganglionic neuron. So we've got a pre-ganglionic neuron, and then a post-ganglionic neuron is short on the parasympathetic nervous system. And then in the sympathetic nervous system, you have the preganglionic neurons, which are relatively short, and then a longer postganglionic neuron running to the specific organs. Okay, so again two, but the arrangement's different. So short pre-ganglionic and long post after the ganglion, postganglionic neuron. Okay. Again, the postganglionic neurons will be non-myelinated, whereas the preganglionic ones, as mentioned already, they have less. I think they have less myelination on them. Okay, some of these motor neurons here can actually go to things like the adrenal glands and actually cause release of hormones into the blood. So that's different as well. Okay, so let's have a look at the different things. So these two systems are antagonistic. So you might have heard that term before. They work against each other. So if the level of activity of one is increased, and that means more nerve impulses coming down the parasympathetic nervous system, there'll be fewer coming down the sympathetic. So the level of activity regulates up and down, and then the neurotransmitters they secrete will affect. So the neurotransmitters are the chemicals they secrete at the end of the neurons and will affect the activity. So you've got this of the organs. So you can see here we've got the heart on both sides. So even though there's electrical impulses coming down here, what's being sent out at the end when it reaches the heart, the neurotransmitter being released here and here, is different, and they have different responses and different effects on the heart. And you're going to look at that when you look at the control of the heart rate. So parasympathetic then, okay. So parasympathetic nervous system is to do with relaxation. Okay, it's almost the opposite of what you think. Sympathetic, you think and parasympathetic, you think that's against it. All right, in my mind, but it's actually the opposite. So it's constriction of the pupil. So, but keeping simulate flow of saliva, slowing the heart rate, constricting the bronchi, so less flow of air into the lungs, stimulate peristalsis and secretion. So you can see, not stressed, relaxed, digesting your food, okay, stimulating release of bile, again, digesting your food, allowing you to go to the toilet, Okay, whereas the sympathetic nervous system is effectively the opposite. So you've got dilation of the pupils, so letting more light in, so time of stress, time of activity, needing to see what's going on around you. Inhibiting the flow of saliva, so this is switching off the activity of digestion. Accelerating your heartbeat, dilating the bronchi, remembering allowing more air to get into your lungs, allowing easier ventilation. Inhibit peristalsis and secretion, so again toning down, switching off digestion, but increasing the activity of the liver in terms of breaking down glycogen stores into glucose, so providing glucose for respiration, for a time of activity. Also secretion of adrenaline and noradrenaline. Remember, adrenaline is that stress hormone, okay, for a time of fight or flight, and inhibiting bladder contraction, so stopping you going to the loo. Okay, so two antagonistic systems, okay, working against each other. You've got a similar table here with information that you might want to annotate onto your diagrams of the layout of the autonomic nervous system that you've got on your worksheet. Okay, and that takes us to the end of the session. Thanks.